This is Xianglun Mao from Biomedical Imaging Research Institute at Cedars-Sinai Medical Center. The topic of this presentation is 3D free-breathing non-ECG T1 T2 B1 plus cine mapping with cardiac MR multitasking. As we all know, quantitative cardiovascular MRI has the potential to perform a wide range of diagnostic measurements in the heart. It can characterize the myocardial tissues, provides an accurate assessment of heart function and anatomy. It can diagnose and monitor the cardiovascular diseases. The NMR relaxation constants T1 and T2 is promising for cardiovascular tissue characterization, and it can be used for revealing some diseases such as fibrosis, edema, myocarditis, inflammation, and even the microvascular obstruction and myocardial infarction. Conventional myocardial T1 T2 mapping have several major technical challenges. They often apply a mixture of breath holding, ECG control, and short acquisition bursts. The breath holding strategy does not work properly for some unhealthy subjects who have cardiac arrhythmias or difficulty holding their breath. The ECG triggering or gating depends on patient's heart rate and often need to sacrifice imaging resolution. The respiratory navigators has been used to eliminate the effect of the respiratory motion during the free breathing data acquisition, but the scan efficiency is low. Our solution here is a quantitative multidimensional imaging framework called MR Multitasking, which uses a low-rank tensor image model to exploit the spatial temporal recollation between images, and it can improve the scan efficiency. It can capture the cardiac respiratory motions, T1 and T2 recovery. It is a single continuous scan that not only provides three quantitative maps, T1, T2, and B1 plus, but also the qualitative maps such as cine images. The underlying image can be represented as a five-way tensor with one spatial dimension and four time dimensions representing four dynamic processes. With a low-rank tensor image model, this large memory extensive tensor can be factorized into five factor matrices with much smaller size, where each of the matrices contains the basis functions characterizing that dimension. And these factor matrices are weighted by a very small core tensor. The degrees of freedom is greatly reduced with such image model. The 3D multitasking sequence is designed by cycling through several T2IR preparations with different duration. 3D flash pauses fill the entire recovery period for the data acquisition. Please note that the flip angle for the 3D flash pauses are alternating for every other recovery period, enabling the B1 robust T1 mapping. Two sets of K-space data are collected for different purposes. The subspace training data are frequently collected at the center K-space line for every 8 readouts. It has a high temporal resolution and can be used to determine the temporal features. The imaging data are collected following the stack of stars K-space trajectory. It covers the entire ventricle and it has a high spatial resolution to determine the spatial features. We validated the multitasking sequence with a 16-mile NIST phantom along with the reference T1, T2 scans. We also scanned 2 healthy volunteers and 3 female farm pigs with reperfusion injury in the myocardium. Both multitasking sequence and the reference T1, T2 scans are collected in those scans. Here's a summary of the important parameters of the multitasking sequence. Basically, we can have the whole ventricle scan with a resolution of 1.4 by 1.4 by 8 millimeters within less than 10 minutes. Here is the phantom result. The multitasking T1, T2 maps are in the left column. The MOLI T1 and T2 prep flash T2 maps are in the middle column. And the conventional IR-SE T1 and SE T2 maps are in the right column. The T1, T2 values in different wires across different methods are comparable, suggesting that the high accuracy of the multitasking method. The statistical analysis further shows that the multitasking indicates a good reliability. and equally good or even better performance in terms of bias and the limits of agreements compared to MOLI and T2-prep flash methods. Multitasking allows us to individually look at each dynamic process at different locations of the multidimensional tensor. For example, different T1 weightings during an inversion recovery process, different T2 weightings, and cardiac and respiratory motions separately. This is an example of a female healthy control age 45. The first row are all the results from the multitasking sequence, including black blood cine images, T1, T2, B1 plus maps at 3 different slices at both systole and diastole stage. They are all acquired using one single continuous acquisition. The second row are the reference T1, T2 maps. The myocardial T2 maps between the multitasking and the references are very comparable. As for T1, the MOLI is known to underestimate the T1 values. Multitasking T1 values are closer to the reported values from Sasha at 3T, which is known to be a more accurate method. This is an example of the pig weighted around 35 kilos. The first rows are all the results from multitasking. The second rows are the reference cine and T1, T2 maps. The reperfusional injury can be clearly seen in the T1 maps, both from multitasking and the MOLI. The statistics also suggest that the injury region has lower T1s compared to other regions in the myocardium. However, there's no significant difference in terms of the myocardial T2 maps. Future work will focus on improving the image quality and spatial resolution and involving larger subject cohorts for the clinical validation. As conclusion, we developed a 3D whole ventricle multitasking sequence with free breathing and non-ECG technique. It further shows that a 3D whole heart is feasible. This 3D multitasking sequence could capture 4 different types of temporal dynamics, and it has the potential to provide T1, T2 cine in one scan, even at high field. Potential applications include comprehensive tissue characterization in the whole ventricle. Since we are able to provide both quantitative and qualitative maps altogether, this technique can be used to replace conventional MR scans for the routine clinical practice in the future. Lastly, let's acknowledge our grant support and the great contribution from our colleague, Dr. Ivan Kotik, for the help of the animal study, and thanks to all the BRE colleagues and Siemens colleagues in our facility. Thank you.